sorry about that. The title for my research and talk today is Investigations on the Role of the Molecular Chaperone Prefoldin in the Progress of Alzheimer's Disease. And I want to accept for my research, I want to give an introduction containing the background with the proteins and folding, misfolding and misfolding diseases. And I also want to talk about the role of molecular chaperone in protein misfolding disease. I will give a background of Alzheimer's disease, and you might have heard many things already by the previous talker, Dr. Nath. And I will go to the ends with my study and also explain my recent results. So, what are proteins? Proteins are particles in our bodies performing all the actions like transport of nutrients, defense against poison, transmission of nerve signals, and catalysis in the body. And we have around 30,000 of them circulating, and the information about every single of it is stored in our DNA. The structure of the protein, the main motifs are either alpha helix or beta sheet, and in the major protein, alpha helix and beta sheet are combined and with interactions and loop. So it starts in the nucleus in the DNA, where the DNA is transcribed into messenger RNA, which is transported into the cytoplasm and translated to amino acid string, and then the amino acid string is folded to the major protein. So basically, there are three important steps: transcription, translation, and folding. And first, when the protein has been folded, it's active and can perform its mission in the body. So what happens if the protein cannot fold? Then it can be misfolded. And misfolding leads to the loss of the natural function of the protein, but it can also lead to gain of a toxic function. And then we're talking about misfolding diseases. So, misfolding diseases, it's the common name for human diseases associated with the presence of fibrillar protein deposit. So in some cases, this happens because of mutations in the precursor protein's DNA, but in most cases, there are other causes, like the previous talker already had talked about, like the lifestyle, environment, food, whatever. But the common thing is the final product of misfolding, which is fibrillar structure composed of the precursor protein, but they have adopted a different shape. They are reaching beta sheet structure. So, to summarize, the bright side of the protein is that they are very important. They are doing all the actions in the body, and we have many of them. For example, insulin regulates the metabolism of carbohydrates and fat, and we have beta-2 microglobulin, which is important in the immune system. Lysozyme is also important in the immune system, and we have OES, which is a chaperone, which has importance in folding on other proteins. So these are a few examples. But we also have a dark side of the protein, and that is the misfolding mechanism that they can do. And then this, this protein end up in modified structures. And also, as you can see, depending on the precursor protein, the final fibril structure can look a bit differently. For example, insulin gives rise to long needle-like fibrils, whereas lysozyme gives rise to more noodle-like fibrils. But they all are involved in diseases. And here are a few known misfolding diseases, except for Alzheimer's. We have Parkinson, prion diseases like mad cow disease, diabetes type 2, and Huntington disease. And these deposits the deposits can be accumulated in different parts of the body. In the case of Alzheimer's, the deposits are found in the brain. But in other diseases like LAS, the peripheral nerve system is affected, and in other diseases, heart and kidney can be affected. So in Alzheimer's disease, now you might recognize a few things that the previous talker talked about, but it's a serious disease. Around 29 million people are affected in the world, and it's the major cause behind dementia. The main risk of developing disease is aging. And to date, there is no disease-modifying treatment available, but there are, of course, medicines that improve the life quality of the patients, but we need to find the missing pieces in understanding the mechanism of Alzheimer's disease. So the main symptoms are memory loss, cognitive decline, mood swings, personality changes, and loss of recognition of loved ones. So Alzheimer's disease is not only serious for the patient with the disease, but also family and friends. So inside the brain, this recognize the brain of Alzheimer's disease patient shrinks dramatically, and inside the nerve in the brain, between the nerve cells, there are protein deposits, and also the nerve cells die away. So in Alzheimer's disease patient brain, there are very few nerve cells and also synapse loss. So to diagnose Alzheimer's disease, some characteristics, there are aggregates consisting of Amyloid-β protein between the nerve cells. Inside the nerve cells, we found aggregates of another protein called tau. And the synapses and neurons are lost within the brain of Alzheimer's disease patients, and also we have inflammation response.